Hello everyone. Welcome to the material for chapter 16. In this chapter, we're going to cover several topics from DNA structure to how DNA is replicated, and then how DNA is packaged into the chromosomes that we're used to seeing on various images. In previous chapters, I've made a podcast that covers the whole chapter. And we've heard from some students that these are a bit long, so what we're going to do is we're going to break each chapter up based upon objectives, and that way you can just go to a particular objective, and that might make your studying a little bit more focused. So the first objective, which will be the focus of this podcast, is on DNA structure. Now, in this objective, it begins with asking you about the evidence for DNA as the genetic material. All you need to know on that is what it says in the study guide. But you do need to know that. There's bound to be at least one question on it. We won't get into the details of how those experiments were done until you get into genetics, biology 326. But I think it's good for us to begin with a basic nucleotide structure, very similar to what you saw several chapters ago when we looked at macromolecules. Remember, a nucleotide, let's draw it over here in this corner to give ourselves a lot of room to work around with, begins with a sugar. I'm not going to draw all the atoms, of this sugar, but I'm going to draw some of them. This is an OH group, a hydroxyl group. And then up here we have a CH2, and very importantly attached to this CH2 is a phosphate. And this phosphate is highly negatively charged, and that's what makes DNA, by and large, a very negatively charged structure. So this is the sugar. That's the first component of a nucleotide. The second component is the phosphate, and then the third component, we'll draw over here, is a nucleotide base. And what we're going to do is we're just going to draw that as a box like that, and put in B for base. It's a nitrogen base, but we're just going to put B for base. We're going to come back to these bases, what they are and how they form bonds. But I'm going to begin this topic now of the backbone of DNA. What is the DNA backbone? And all it is, as we remember from earlier chapters, is a repeating unit of this monomer, the nucleotide. And so what happens is the next nucleotide will bind to this hydroxyl group on this nucleotide. Okay, so let's call this nucleotide 1 for lack of a better way of calling it. And then we'll draw our second nucleotide here. And so this is going to bind to a phosphate. So I'm just going to draw PO4 for phosphate. We also, in this process of forming this bond, we're going to lose this hydrogen. So I'm just going to scribble this H out, but we keep the oxygen there. Okay, so let's see if I can draw this reasonably well. We have our CH, like so, and now this is going to be attached to our sugar. It's not a beautiful drawing, but you probably didn't expect much more from me. So this is our sugar of our next nucleotide, and it also has an OH group on it. And it's going to have a base attached to it. Now let's, for fun, just draw a third base pair. So here's our third base pair. We'll draw the sugar first. We have our CH2 right here. I forgot this was a CH2 here. And then we have our phosphate group here, which binds to this hydroxyl group, and remember we lose this H here. Okay. And we end up with this hydroxyl group right here. So this is our DNA backbone. Now, a piece of DNA is going to be thousands of these nucleotides long, but we're just showing 3 nucleotides here. I would say you should be familiar with looking at this, a structure like this. To learn this, the best way to learn all of this, and we really encourage you to do this, is to draw this out. And as you're drawing it, you're going to remember it a lot better. We promise you, that'll work. And who knows, you may have to draw it on an exam as well. But in no more detail than I've drawn here. Forgot to add our third base here. Now, a few things I want to point out here before we move on is to remind you that we have that phosphodiester bond. And that phosphodiester bond forms between this phosphate group and the oxygen. We've already learned this, but it's important to kind of review it again. And because of space issues, I'm just going to write P-D. But know what I'm saying here is that right here is that phosphodiester bond. There's also a phosphodiester bond right here too. So this molecule I've drawn has two phosphodiester bonds. I also want to talk about the polarity of the DNA molecule that we see here. DNA, when we write it, we always write it in a 5' to 3' direction. Now what do I mean by that? Well, let me explain that. This end of the DNA molecule we call the 5', and I'll explain that in a moment. And this end of the molecule we call the 3'. For the purpose of this test and the material we're covering in this class, that's what you need to know. The phosphate at the end is the 5', the hydroxyl at the other end is the 3', and we always write DNA in a 5' to 3' direction. If you're curious where it came from, and so you're one step ahead when you get to genetics, the reason this is called the 5' phosphate is because sugars are numbered 1, 2, 3, 4, and then 5. So this phosphate is attached to the 5' carbon. And likewise, remember I just called this a 3 here. This hydroxyl group is attached to the 3' carbon. That's all we mean by it. But again, just know that this is the 5' end and this is the 3' end for this test. Alright, now I want to introduce another topic. We just talked about how that DNA molecule on the last slide formed this backbone of DNA. So let's really briefly draw what we had on the last slide, but in a lot less detail. Remember, we had our base here, and we had our sugar, our ribose, here, or deoxyribose, depending on what we're talking about. And then we had our phosphate. And then remember the next nucleotide base was here, with the sugar attached to it, and the phosphate. And then this phosphate formed the phosphodiester bond with this sugar. And then we had our third base here. Let's go and draw that just so it looks similar to what we had on the last slide. And just to be consistent, remember we called this 5' to 3', and we had our bases, 1, 2, and 3. When we see a picture of DNA in the media, and newspaper, wherever, we always see it drawn as this double helix, with one strand here, and then our other strand like so. So how does this other strand bind to this one DNA molecule here? It does so using these bases that we haven't really talked much about. So we're going to have a base over here, also attached to a sugar and a phosphate, and then another base shown here, attached to the sugar and then the phosphate. And just like we saw on the other strand, the sugar and the phosphate form these phosphodiester bonds. And let's draw this third one just to complete it. So how do these, well, before I talk about how these bind, there's another really important concept that we have to mention here. It's on your study guide, and so I want to make sure you understand it. DNA is what we call, let's draw it up here, it is called antiparallel. And what we mean by antiparallel is that this strand is written in the 5' to 3' direction, and in order to form this double helix in the proper structure, it's written in the reverse way. So this end down here is, let me make that in red. This one down here is the 5' phosphate, and then up here we have our 3' hydroxyl. So that's all we mean when we use the term antiparallel. One strand is written in 5' to 3' direction. The other strand that forms the DNA double helix is written in the reverse orientation. 3' at the top, 5' at the bottom. That's all that means. Okay, so now let's go back to how these nitrogen bases on the nucleotides form hydrogen bonds between each other. It's a pretty important concept. I think first what we need to do is talk about the different nucleotide bases we have. DNA and RNA each have 4 bases. There's a few differences, but for right now, let's just say we have these 5 bases. We have guanine, we have adenine, I'm just writing the first letter, guanine, adenine, we have thymine, and we have cytosine. These are what we find in DNA. RNA is very similar. In fact, why don't we just go and write this over here? DNA. RNA is similar. We have guanine, we have adenine, we have cytosine, and instead of thymine, we have uracil. So these are the 4 bases of RNA. For the moment, we're going to focus on DNA. These bases here, we'll put our bases in these letters here. So let's just go and write some here. We'll put a G here for guanine. We'll put an A here for adenine. And we'll put a T here for thymine. Now we know automatically, because a set of rules that were discovered about 50, 60 years ago, what these have to be. And that's because we know that guanine forms hydrogen bonds only with cytosine. Adenine only forms hydrogen bonds with thymine. And of course the reverse is true too. T will bind to A, just like so. Now I said they formed hydrogen bonds, so let's go and draw some hydrogen bonds here between these. Okay, as you see, as I drew these, I purposely drew 3 sets of hydrogen bonds between G's and C's and 2 between A's and T's. And that's because when these bonds form, they will form very specifically 3 between G's and C's and 2 hydrogen bonds between A's and T's. In your note-taking guide, it asks a question: what kinds of bonds are stronger? And we know, because G's and C's form 3, that nucleotides that are held together between a guanine and a cytosine, that's a stronger interaction than between A's and T's. And we're going to come back to this point in a moment, the strength of these bonds, but for right now you should recognize that G to C bonds are much stronger than A to T bonds. Okay, now I want to come back to these bases, because in your note-taking guide it asks you to tell me which ones are purines and which ones are pyrimidines. And so I think what we're going to do is, why don't we just make a little box here. And you can draw it somewhere on your paper as well. Don't worry about this scratch off here. Made a slight error. So we have pyrimidines. So let's go and write the word pyrimidine. And in addition to pyrimidines, we have purines. So we'll write the word purine here. Our pyrimidines are cytosine, uracil, and thymine. Our purines are guanines and adenines. Now I often come up with weird little ways that helps my mind remember what these are. And so I always remembered this, that CUT spells the word cut. And if I think of a pyramid, I think of a pyramid as having a very sharp tip to it, and that could cut you. So that's how I always remember that cytosine, uracil, and thymines are pyrimidines, because of the word cut. And then the other two, I just remember the ones that are left over. GA are the purines. Thinking of ways to remember different things, I should have mentioned this earlier, but while we're thinking of it, the way I always remember that G's bind to C's and A's bind to T's is because the letter G, the capital letter G, and the capital letter C have this curve, a similar curve to them. And so I always remember those are the 2 nucleotides that bind. And A's and T's are written with straight lines, and so those 2 bind. Again, just kind of a weird way I remember things. If it helps you, great. If it doesn't, ignore it. Okay, now that we've gone over the rules for the structure of DNA and talked about how it's formed with the different kinds of bonds, I have a little problem I want you to work on. And in this problem, I'm not going to tell you the answer. I want you to work on it on your own, and you can talk to it with classmates. But if you can understand this problem, you'll really understand most of what there is with this first objective. So I'm going to write the first DNA molecule here in red. And then I'm going to write the next DNA molecule down here in blue. And remember, these are 2 separate molecules. And let me go ahead and tell you that this is the 5' end here of this molecule, and the 3' end at the other end. And over here the same thing. This is the 5' end, and then the 3' end. Now what I want you to do is, underneath this strand, I want you to write its complementary bases. You can draw it in this little box if you like, or you can just draw them down right below it, whatever works for you. When you write this, you need to remember the complementary base pair rules, which bases are going to bind with which bases. You also need to indicate what ends these are. Is this the 5' end or the 3' end? Remember our rules of antiparallel. So write the complementary bases, and then I want you to answer a few questions. For each one, how many base pairs are present in this molecule, and how many are present in this molecule? So I'm just going to write BP for base pairs, but I want you to count the number of base pairs. This is good practice. Next question I want you to answer is, how many phosphodiester bonds? And for simplicity, I'm just going to write P-D for phosphodiester bonds. How many phosphodiester bonds do we see in this molecule? How many do we see in this molecule? Then I want you to answer, even though we don't see the sugars, you have to recognize that there are sugars attached to each of these nucleotides. So sugars, how many sugars? And then answer that question. Then I want you to tell me, is this molecule, both of these, you should be able to answer this question here, is it DNA or RNA? And I'll give you a hint. They're both either DNA or RNA. And then finally, what I want you to tell me is which one is going to be harder to pull apart, meaning which one forms stronger hydrogen bonds with each other. This one here, this molecule here binding with this molecule, or is this stronger, this red one binding with the one you draw below it? Which one is stronger? So, is the blue one or the red one stronger? And you should be able to explain why that's the case. So again, write the complementary base pairs down here with the correct polarity using your antiparallel rules. Then I want you to ask, how many hydrogen bonds are there? I wrote base pairs here, but that's just counting. That's not as difficult. Let's take that off. I'm not sure what I was thinking there. How many hydrogen bonds are there? That's a better question. That's a more respectable question. How many hydrogen bonds are there in each of the molecules? Sorry about that. You can tell me how many base pairs there are. That's cool too. But really what I want to know is how many hydrogen bonds there are in this molecule and how many are in this molecule. Then, how many phosphodiester bonds do you see in this entire molecule? And how many do you see in this entire molecule? How many sugars do you see in this one and this one? Then, are these molecules representatives of DNA or RNA? And then finally, which double helix is stronger, this one or this one? Again, if you can answer those questions, you understand this particular objective very well. Okay, if you have any questions about this, please let me know. And make sure you come to the next podcast, which will be on how DNA is replicated. All right, I'll see you in class. Bye.